Thanks, Daniel, and thanks, Bruce, for inviting me. I have a heavy burden here, and I'm quite anxious, which I usually am before a talk, but in this case, since I'm apparently the only, or one of two non-Boston speakers, it's a lot to live up to. And now Keith told me I had to be a Rolling Stones act and explain everything. So to extend the metaphor, I guess I have to say, sing a few bars of "You Can't Always Get What You Want." If you try, you get me. So I want to thank Keith for great data, great work, and great intro. I guess I'll start by saying very quickly what you all know. ERK is supposed to be activated in lots of human tumors. It's supposed to drive lots of human tumors. There's at least 3.5 common mechanisms of activation: RTKs, mutation in K and NRAS, or mutation in BRAF. And I don't, Keith, did you mention that NRAS is mutated a lot in melanoma too? 20–30%. So, and Karen Chakowski's not here, but I'll at least say that NF1 is important and probably more important than we thought. In some of those tumors, it works by activating ERK. So we don't trust genetics. We're too lazy to do siRNA, so we like to use drugs because they have lots of other advantages, like they can tell you about output and they can tell you about dynamics, which I'll talk about a little later. So in 2005, David Solit did this not very sophisticated experiment. He took the Pfizer MEK inhibitor, well, the Park-Davis-Pfizer allosteric MEK inhibitor, which is quite selective for MEK. He took lots of cell lines which had activation of mutated EGFR or amplified HER2, which was the RTK activation model, mutated NRAS or KRAS, and mutated BRAF. And this is a very famous high-impact journal paper, basically because of a very prosaic result. All the RAF mutant tumor cell lines are sensitive, all of them, except for 2 out of over 100 now. The only 2 that are completely resistant have RB loss. The rest are sensitive in some shape or form. As Jeff would predict, some undergo lots of apoptosis, some don't. Some don't even stop growing, they grow slowly, and we wouldn't think that's very clinically important, but they're all sensitive. They all slow down at least a lot, usually completely, sometimes with apoptosis. Lots of these have PTEN loss. They are almost all sensitive, but they don't undergo apoptosis. If you look at RAS tumors, some are sensitive, some aren't. I'm not going to go through why today, but lots of RAS tumors retain lots of MEK dependence on ERK signaling. The surprise, which as usual is almost what the paper got the paper not accepted as opposed to being an interesting surprise, is the RTK tumors. And I don't think this goes for all RTKs, but it goes for mutant EGFR and amplified HER2, are insensitive. They're insensitive in vitro, they're insensitive in 3D, they're insensitive in vivo. I'm going to get back to that later, but I'm going to jump right into RAF. So the original MEK inhibitor studies with the, in clinic, I mean, Solit and I really trumpeted this. This is back in 2005. The original MEK inhibitor studies were not negative, but they were disappointing. There was about a 10% response rate, short-lived responses. So the question was, why? So since it's Bruce's symposium named in honor of Bruce, I'll give you the, but I think is the punchline later. One possibility was, are non-MEK effectors of BRAF important? As Keith said, I'll get to that in a minute. And RAF does have other substrates, and we're not inhibiting RAF with the MEK inhibitor, we're inhibiting MEK, but I'm not going to show the data. I will tell you so far we have no data that the biologic and biochemical effects, downstream biochemical effects of MEK and RAF inhibitors they're almost overlappingly identical. Do coexistent genetic and epigenetic events, stem cells, tumor heterogeneity, all the things we usually say when we fail, including my favorite, the tumor cell is really smart and gets around whatever we do. But there's a third one that old guys like my mentor Bruce and I think about, which apparently nobody else thinks about in this advanced day and age, is target inhibition adequate? What's the pharmacodynamics? As Keith pointed out, you get you do get pretty good inhibition with a MEK inhibitor, but you inhibit everywhere. And you have a dose-limiting toxicity, which is rash. So for all the 18 million papers on what ERK does in all sorts of systems in the cell, what you get in patients is a rash. It may be boring, but that's what you get. So the question is, does the therapeutic index limit the amount of pathway inhibition? Now, no one measures the pathway inhibition quantitatively well, but you're probably getting 60 to 70% inhibition with MEK inhibitors. So we decided to try RAF inhibitors, and Keith knows I'm going to say this, so I'm going to say it. Everybody gets this wrong. They are not selective BRAF inhibitors. None of the inhibitors so far extant is selective for BRAF. Now, or at least they are all ATP competitive inhibitors. They bind the ATP binding site of each of the 4 enzymes that we're going to talk about, wild type A, B, and C RAF, and the V600E. Now, this is also operational data. It's flawed or limited by the fact that it's very difficult, pretty much impossible to purify holoenzyme, probably because it requires co-purification with chaperones. But if you take the catalytic domain, the truncated purified catalytic domain, this is Gideon Bolag's data from Plexicon circa 2006, it inhibits mutant B and wild type C almost identically. It's about 3-fold worse, a little worse, but not extraordinarily for wild type B. And we did this experiment. I don't think anybody else did it, which sort of, surprised me. But if you just IP, A, B, C, and V600 and do it in knockout cells, so you get only the one you're looking at, you basically inhibit all of them over approximately the same concentration range in vitro. This is in vitro IP. So we did the same sophisticated experiment in about 2007 and got a surprise because we thought sensitivity to MEK inhibition implied dependence on ERK. So we got the same sensitivity of all the RAF cells. The difference in the bars are higher because the Plexicon drug is less potent. This is concentration is the ordinate. We got the same insensitivity of RTK tumors and of the ensemble of RAS tumors that are resistant to the MEK inhibitor. But all of the RAS tumors that are sensitive to the MEK inhibitor are insensitive to the RAF inhibitor. So this was a surprise. Why was this? Was there something funny about MEK inhibitors? So we solved this conundrum in about 72 hours, and here's the key piece of data. When you give a RAF inhibitor to a cell with V600E BRAF mutation, you get exactly what we all would have thought. You inhibit phosphorylation of MEK, you inhibit phosphorylation of ERK. That's what's supposed to happen. When you give it to any other cell, you activate the pathway. You activate MEK phosphorylation, you activate ERK phosphorylation, you transiently activate transcriptional output of the pathway, which I'm not showing here. So this gave me a huge headache because I thought if there was one thing I understood, it was that RAF activates, MEK activates ERK. And this one took us about, we discovered this in about 2007, and it took us, or 6, it took us 3 to 3.5 years to solve. So this is just a summary of Keith's talk in an interrogatory way. Why use this drug in patients? It might make tumors worse. You're going to have to carefully genotype anyone. I mean, we should do that with everybody now, but in this case, you've got to be real sure. But there should be no toxicity due to inhibition of ERK in normal cells because it doesn't do that. You may get toxicity due to activation, and that's what Keith showed you. And this could increase the therapeutic index and allow higher dosing and better pathway inhibition and increased efficacy in RAF tumors. Okay? Now, this is supported by Bolag's data in the phase 1 that you don't get responses. He tried quantitating as best he could, but you have to take the number with a grain of salt. You don't get responses if you inhibit the pathway less than 80%. So I'm going to have more courage here than Keith, and despite crizotinib, stick to my, my point that you try to make selective drugs because your ability to inhibit the pathway effectively is going to be limited by off-target toxicity. On-target toxicity, that's what the enzyme's doing, and you're stuck. But off-target toxicity, you're not. And I think crizotinib will be overridden by drugs that many of us know about now that are more selective. Okay, so let's get back to my migraine. How can an ATP-competitive pan-RAF inhibitor activate RAF kinase and ERK signaling? So, and why, and we were so stuck on that, we forgot the flip side. Given that there must be some mechanism for this, why does it inhibit ERK signaling, but in V600E cells only? So there's a paper we wrote, a guy named Poulikos did this work in my lab. He has, in collaboration with Chao Zhang and Kayvon Shohat's lab. It's a Nature paper last March, and to get through, I always spend years discussing this, so I'm just going to show you the PowerPoint cartoon, and you're going to have to presume it's right, and then I'm going to show you what causes resistance. And what causes resistance is based on the mechanism. So the first thing to say is it a selective RAF inhibitor, BRAF inhibitor? No. If you do BRAF knockout cells, if you get MEFs, you activate just as well. And remember, activation occurs in normal cells that don't have mutant BRAF. So it ain't, it's Boston, it isn't a selective BRAF inhibitor. Okay, is it an idiosyncrasy of the Plexicon drug? Every drug we've seen does it. And what it does actually is you get an interesting dose-response curve. As you go up in dose, at the dose that approximates binding to the site, you see induction of ERK phosphorylation. When you increase the dose a lot, you start seeing inhibition. So there's a parabolic dose-response curve, if I can say that, biphasic if you like. And this should have given us a big clue, which of course it didn't because we're only MDs, but this is what we believe is going on. The way you activate RAF physiologically and in RAS mutant tumors is in a RAS-dependent fashion. There is a domain in RAF that inhibits its dimerization. RAS also binds to that domain, and this is going to be important later, and the binding of RAS to this inhibitory domain causes RAF to dimerize, okay? So RAS is inhibiting the autoinhibition of dimerization of the molecule. You get dimerization, and that and several post-translational modifications by phosphorylation cause activation. We know this because deletion of the amino terminal autoinhibitory domain causes constitutive dimerization and constitutive activation. In a cell with enough RAS activity, and this can be a normal cell, but the higher the RAS activity, the more dimers you have, binding of the drug to one site to one of the members of the dimer allosterically activates the other. And that's what's shown in that paper by chemical biologic techniques. That allosteric activation is associated with negative cooperativity of drug binding, so that it is hard to get, you need much higher concentration to bind both and get inhibition. This half-bound dimer has about a 30-fold increase in specific activity, activates signaling, and is responsible for induction. Now here's the weird part, or at least what I thought, again, not being smart, I thought it was the weird part until we figured out why. Why do you inhibit in the tumors with V600? Well, it turns out that in melanomas with RAF mutation, RAS GTP levels are extraordinarily low. So you have monomers. Now this is a sticky point in the mechanism in the original paper because RAF is not thought to function as a monomer. But nevertheless, you have monomers, you have low RAS GTP. The great majority of mutant BRAF in those cells is a monomer. The drug binds, and you get inhibition. So, and this is consistent with the experiments we did where you put mutant RAS into a cell with mutant BRAF, either a 293 experimental model or into an actual melanoma or colon with mutant B. You put in RAS, elevate RAS GTP, you create resistance to the RAF inhibitor but not to the MEK inhibitor. Stick in HER2, increase RAS GTP, same thing. Now resistant to RAF inhibition but still sensitive to MEK inhibition. So given all this, and given what Jeff told you, and given what everyone else has been finding, starting with Charles Sawyer's, how could you get resistance? And I'll go through this real quick, and I'll tell you what's good and bad about these, what's true or not, but here's theoretically what you'd think. First of all, you could get gatekeepers. As Keith didn't directly say this, but it's what he said when he answered the question, no gatekeepers have been found so far in patient tumors, even though gatekeepers exist that you can get to with a one nucleotide change. So why is that? I don't know. Levi Garraway has reported that activating mutations in MEK1 that confer MEK resistance can also confer RAF resistance. I think that's still work in progress. Levi has shown that the COT kinase, which is a MAP kinase that would be a so-called Engelmann bypass lesion, can cause resistance in culture. I think right now we have to say that's work in progress because I'm not sure that's been convincingly demonstrated yet in tumors. And it has to be said, not enough resistant tumors have been examined. And Dave Solid at Sloan Kettering and Mike Davies at MD Anderson have shown that PI3K-AKT activation, that PTEN loss makes the tumor less sensitive, but they're still sensitive. So how that may be a way of enhancing effects, but I don't think it causes resistance. So the other things that could cause resistance are anything that activates RAS-GTP. Mutation in RAS. And mutation in NRAS has been found in a few tumors, and at the end of the talk, Keith can tell you how many, I don't know. But it's a very small minority, but mutations in NRAS have been isolated. And there are two receptor tyrosine kinases for which visible papers have been published as they cause resistance, PDGFR, which I can say there's absolutely no evidence for, and IGF-1R, for which I can also say there's absolutely no evidence for. We can argue about this later. I'm from New York, after all. But I think most people agree with that, although I think they are plausible mechanisms of resistance because they ought to activate RAS-GTP. Now, what we miss completely, and the interesting thing about these resistance mechanisms is they're site-specific. And I'm not talking about the protein, I'm talking about the institution that discovered the mechanism of resistance, and I won't go into that further. So what about the theory about how the drug works? Well, we basically say the drug works because there's low RAS-GTP in the cells and RAF can't dimerize. So what the model suggests, and believe me, I'm sure there's many things that are going to go beyond the model and make it better, but the model suggests anything that activates RAS-GTP, but also any RAF mutation that causes it to dimerize in a constitutive manner. So Dave Solid and I were jealous of all the people who were looking at resistance in cell lines, so we did the same thing. It was slightly different in that we cloned a sensitive line into a high dose, a clinically, the dose to which a patient is exposed rather than small increases through time. We got 5 clones independently isolated from one particular BRAF mutant PTEN wild-type cell line, and being too poor to do genomics, epigenomics, metabolomics, we did a sophisticated experiment we've worked at Sloan Kettering called a Western blot. And on the Western blot, we found that 3 out of the 5, actually these don't have gatekeepers, they don't have any of the things I've talked about before, but they do have a 61 kD truncated RAF, 3 out of the 5. I'll try to do this fast. This is going to come out in Nature sometime. These clones are RAF inhibitor resistant. They are MEK inhibitor sensitive. And again, the general theme, Jeff beautifully described the complexity of resistance, but the first resistance you seem to get is resistance that prevents the drug from inhibiting the pathway. So these, you have an altered RAF, you can no longer inhibit the pathway with a RAF inhibitor, you can still inhibit it with a MEK inhibitor. I'm not going to show it, but you transfect it into cells and you make them resistant. So we cloned RAF, and I should say these cells also are heterozygous for the RAF mutation. They have a wild-type copy and a mutant copy. What we found in the resistant cells was you have a full-length message which is wild-type now completely. This is the parental, you probably can't see it. And you have a new shorter message which confers, which we cloned and we analyzed, and we basically found that it constitutes a splice, a loss of exons 4 through 8. Okay? I know everybody's going to ask me. I don't know what generates the splice rigorously yet. There is a cis mutation in the, mutation in the mutant allele, not the wild type allele, in the splice acceptor site in intron 9, okay? Now, but we haven't proven that's what confers this. The interesting thing about it is, well, first of all, we've done this in several cell lines and in patient tumors, and every time they're heterozygous, the splice occurs in the mutant allele, which decreases but does not eliminate the possibility that this is a trans loss of splicing fidelity. It strikes us as more, at least in the few examples we've looked at, is probably a mutation. What is interesting, and to cut to the chase, what you're deleting here is the anti-dimerization domain that binds to RAS. I'm just showing that here. It's in exons 4 and 5. So the hypothesis is that V600 signals as a monomer and that the truncated V600E is resistant to the RAF inhibitor because it lacks this domain and homodimerizes and is no longer inhibitable. So from previous work in the first paper, we had examined and mutated the dimerization domain, and we found an arginine 509 residue, and Therian has also done a lot of work on this. The arginine 509 residue is key to dimerization. Arginine 509 mutants of wild type destroy or severely attenuate activity. Wild type and dimerization. Wild type requires dimerization. So I'm going to use an R509H mutant in what I'm going to say in the next couple of slides. So the first things we did is A, V600E. This is MEK activity in a V600 expressing cell, very nicely inhibitable with the drug. Make the 509H mutants, which I guess on the next slide I'm going to show you doesn't dimerize. It still signals, it's still sensitive. So V600 does signal as a monomer. Secondly, if you put into cells V5 and MEK maybe, V5 and flag tagged on different molecules, RAFs, and look at dimerization based on IPing one tag and blotting for the other. In a low RAS GTP cell like 293, you get a tiny bit of dimerization of V600, much more dimerization of the P61, and that dimerization is eliminated by the ARG509H mutation. So here's the cool experiment, at least I think so. I showed you the V600E before. The truncated protein is active. It is resistant to the inhibitor. If you make the truncated protein with the 509H domain, which no longer dimerizes, it's still active. It has a slightly lower specific activity, but it now becomes sensitive to the drug. So if you make the monomer of the resistant truncated RAF, it's sensitive. It's the dimer that creates the resistance. And that's just a diagram. So basically, the way RAF gets activated in human tumors is via dimerization, via RAS activation, which causes dimers, or Arul Chanayan has found several fusion proteins that are rarely present in gastric, prostate, and melanomas. They all delete the same domain. And we've shown that they require dimerization for activity. Or there's one common mutation, V600, that's active as a monomer, doesn't require dimerization, and that's the only class of mutations that is sensitive to these drugs. However, their resistance is mediated by either activation of RAS or a mutation that mediates dimerization. So obviously, one of the things one would like to do is find dimerization inhibitors, which we're actively trying to do. Any time left? Okay, so let's talk about this, which is, I did this with Posse in Japan, with Bruce present. These all look the same, these waterfalls. So this is my take on Jeff's question, and it's an open question. What I'd like to do is shift this from left to right, and then I'd like to see if that makes things better. Now, Jeff has made a great case that is in part a function of BIM, but I would make a great case that or at least a case, that the gradient you see in BIM is going to depend on continued inhibition of the pathway. Okay? So this is what Jose Bazelga and I have been fixated on for the last 6 years. When you activate an oncogene, you get sustained negative negative feedback throughout the signaling network. Okay? Now, if you get that feedback, that constitutive negative feedback, why do you transform? And the idea is that transformation and driving the tumor requires disabling of the feedback by the oncogenic mechanism. So you activate an RTK, you activate RAF, MEK, and ERK, you get feedback inhibition of RAF, you get feedback inhibition of RTK signaling. We've shown that in the HER2 mutant EGFR tumors, you have all this feedback is south of the receptor, it's intact, and despite the PERK, the output you get is very low because of this feedback. How do you disable this feedback? Well, one way is with mutant RAS. The feedback inhibition of RAS is abrogated. You still have feedback inhibition of MEK, RAF through ERK, so you actually activate the output. You greatly activate it, but not nearly as much as you do with V600. Why? V600 enormously activates the pathway because it no longer requires RAS, because it no longer requires dimerization. We never see a RAF mutant that requires RAS-dependent dimerization, and I think it's because you don't select for them. Because if they activate ERK, you lose the dimer, you won't activate pathway that much. You also abrogate the RAF-MEK pathway with this, and you get enormous activation of pathway with really low levels of RAS-GTP. However, when you inhibit the pathway, if all this is true, you inhibit the pathway, there's huge output, the tumor is dependent on it, it starts to die. But you also relieve the feedback, and that ought to limit the anti-tumor response. And Jose and I first found this in a famous figure that I'm showing almost as much as Gallo used to show the HTLV1. That's a private note to Bruce. When you give rapamycin, you inhibit the pathway, but you activate AKT. You give rapamycin to P10 negative prostate cancers, no patient gets through one cycle. And Howard Shear and Dave Sollett and I have found, and Charles Sawyers, you actually activate growth in 40% of the patients, you activate PSA. So what happens in melanoma? You inhibit RAF, you inhibit ERK, you reactivate RAS, you reactivate RTK signaling, you get an induction of ERK. So what happens is when you reactivate RAS, you get RAF dimers, and the RAF inhibitor stops working. That's an overstatement. There's a set point somewhere that I've yet to define. In some tumors, the release of feedback induction of dimers is low, and you get a new steady state, which you see here in a melanoma. This is grossly overexposed. You get a little bit of induction of ERK. In other tumors, like some colons and in thyroid generally, it comes all the way back up. So depending on the feedback set point, the drug's going to work well, modestly, or not at all. And the feedback set point is in part dependent on ligands. So you see this induction of RAS, which is actually to a very low level. It's just overexposed. You get this induction of ERK, which is associated with increased output. If you now, how do I know the drug's not disappearing? If I re-add the RAF inhibitor, this doesn't go away. If I extend this out to a week, it stays just at this level. It's a new steady state. But it's quite sensitive to the MEK inhibitor. And that, I believe, is why Keith's experiments are working so well. But there are other tumors where this comes right back up, and you're reactivating RTK, AKT, STAT3 signaling as well. And to me, this gives a logic for combination therapy that Jose's been doing beautifully with the estrogen rapamycin trial. We believe that's why that works. We're doing in HER2 tumors, but I believe that's the way, since I believe only one thing every 10 years, I believe that's the way you look for combinations. So I won't say anything more, except we're going to propose a new clinical experiment for any tumor. You biopsy the tumor. At MGH, you determine the driver mutation if you can. If you have a drug for that driver mutation, You give a high dose of that drug, trying to inhibit it maximally. But I think what we should be doing at that point is at time X, which I cannot define, but it's not weeks, it's not months, it's at most a couple of days and probably earlier, you rebiopsy and as best we can determine the mechanisms of adaptation. Jose and I call this adaptive resistance, and then the trials are a driver inhibitor plus an inhibitor of some limb of that pathway feedback. So, and the goal is, as I would say, totally agree with Jeff, is to maximize cell death. So that's all I'm going to say. This was all done by other people. People in my lab are Poulikos Poulakakis, Christine Pertilis, Eric Joseph. Strong collaboration with Dave Solit, forever, with Maury and Jose Bezelga, with Kayvon Shochat, with Keith and Jeff Sussman, Paul Chapman at Sloan, and Tony Rebus, who are very, very generous, especially Keith, with giving out tissue. And you can't do this without drugs, and drugs are mostly made by drug companies.
So time for a few questions.
I was wondering if you already started to check whether these splicing variants are present in melanomas before and after treatment with.
I can't believe I didn't say that. I knew I was, as usual, pressed for time because of my loquacity. As opposed to, I was setting myself up for none of these other things have been found in tumors. We find this in 1/3 of the small number of tumors we've examined. The small number of tumors we've examined is not 3. It's on the order of about 20, 21. So I think we have 7 out of 21 with the splice. There was a slide for this. The splice is not always identical. That is, it's always exons 4 and 5 contained, but the splice at the, what's cut out at the C-terminal is different. It can be 8, 9, 8, 10, never the catalytic domain. Early, we haven't done well enough. Basically, this is RT-PCR assays in the tumor. So, like 35 cycles it takes to detect it. We start seeing a smudge in normals at 50 or 55 cycles, whether that's, what that entails, how many cells in the tumor have it. We have to look for in all the cell lines we've looked at and all the untreateds, we haven't found a high amount of splice, but I certainly would guess that it is present before therapy in some substantial portion of the patients.
Bruce?
So, Neil, you just spliced out the bubble on your microphone, but other than that...
Yeah, okay.
No, but do you think in mutant RAS tumors that they're activating by eliminating the inhibition of dimerization of RAF?
No, they're activating the dimerization.
Well, that's what I'm saying. They're eliminating the inhibition of dimerization.
Yeah, that's how it works. Well, that is the cartoon model we all have, the field has about how they work. They bind the autodime inhibition region. Now, I don't think that's all that RAS is doing, but I think to those of you who do this sort of thing, there's an obvious, powerful screen here, cellular screen, since a dimerization inhibitor will not work in these cells, and a RAF inhibitor will not work, but they will work in combination. We also have an amino acid that's required for the dimerization, and we have a post-translational modification that's required for the dimerization. So a dimerization inhibitor ought to work on the resistance, and it ought to work on RAS tumors with the enormously big if of therapeutic index.
Yeah.
So Neil, can you comment again about why it takes so much drug to inhibit the second part of the dimer, and could irreversible inhibitors be the answer there?
Okay, so irreversible inhibitors, this is an allosteric effect. We know that irreversible inhibitors induce at modest concentration. The presumption, since we have not been able to, we are trying hard to get an in vitro system into which to mimic the parameters. As you will all recall, allosteric activation is always associated with cooperativity. So for a while, again, I wasn't thinking very intelligently about it. I thought there was negative cooperativity, as one would find in the Koshland model of allostery for the drug, which is true, but that makes no sense. Why do you activate and get negative cooperativity of binding? Until I realized that this is probably a Michael Eck model. That is, you have a typical Monod-Changeux allostery. You have positive cooperativity for ATP binding. ATP then competes out the drug, and it's a little bit like what you find with mutant EGFR. What you create in the second site is much more ATP avid. We can mimic this with non-hydrolyzable ATP. So what you could do is have it with a more potent drug that bound better than ATP, but if you have a pan-RAF inhibitor, that won't work. You will activate RAF wild-type dimers. So what you will need to do, which may be happening, is a selective mutant BRAF drug of higher potency than what we've seen so far.